Hi, I'm Thomas. Welcome to my research presentation. Watch this. This may look like a computer simulation, but it's real footage from a biological neuron. The video shows a voltage over the cell membrane during an action potential. The fact that we can film voltage like this is pretty significant, and we'll see why a bit. But first, let's zoom out. Take your favorite topic in cognitive science. For any of those, if you want to understand the mechanism behind it, you often have to look at how certain neurons are wired together. And quite often it is also important to know how this wiring changes over time. And to look at the wiring, we have to put the brain under the microscope, which is why we turn to animal models of behavior and mental health. So, how do you find out how neural connectivity changes? You currently have to do something like this. You have an experimental intervention and you want to know the wiring before and after. To find the wiring, you take out the brain, you slice it very thinly, put it under the microscope, and use these images to reconstruct the axons and the dendrites. Now you can imagine that taking out the brain and slicing it up is a pretty destructive operation, which is why you would need more than one animal to do this. The first animal to find the wiring before, and the second animal to find the wiring after the experiment. There are two problems with this. One is that you're not looking at the same brain, so if you find differences in wiring, are they due to the experiment or due to them being individuals? The second problem is that this is wasteful in animal lives, especially if the experiment has multiple stages. Now what if you could reconstruct the wiring in an animal without having to kill it? This would solve both problems at once. You're looking at the same brain every time and you save lives. This idea is what we are going to talk about. So first, some more about this new recording technique, because this is the context that makes our research possible. And then the core of our idea. My supervisor and I, we think we have an idea for using such recordings to find connections between neurons in living animals. We have done some preliminary testing of this idea, so I will show you how we tested it, the experimental setup, and results of these tests. Finally, we take a step back to look at what we've done and what is still to be done. We start with the recording technique, which is called voltage imaging. So the classic method is not to use voltage imaging. Rather, it's to insert a needle electrode through a hole in the skull and poke around till you find a neuron that you can puncture. Though this may sound primitive, it gives pretty clear recordings. You can see not only the action potentials or spikes, but also the sub-threshold voltage fluctuations. That is, smaller voltage signals when the neuron hasn't fired yet. And this technique can be done in living animals, but you can only record from 1 or maybe 5 neurons at the same time. Plus you're poking around rather blindly. So that is the old way. This is a new way. This is a membrane protein that molecular biologists have been developing over the past decades. If you shine light on it, it shines back. And the essence of it is that the amount of light that it shines back depends on the voltage over the cell membrane. So in other words, the reflected light is proportional to the cell's voltage. And that means is that you can use such a protein as a remote voltage sensor, which is exactly what is done. So neuroscientists, they insert this protein into the brain. It nestles itself in all the cell membranes of the neurons, and then they shine light into the brain and look at the light that is reflected back through a microscope. So this is an example image, zoomed in on one neuron cell membrane, shining brightly. If you make, instead of a static image, if you make a video, we can see the voltage fluctuations over time. So the data you're looking at here is from an experiment, quite recent, in which they did both the classical technique with a needle and the new technique with voltage imaging at the same time in the same neuron to be able to compare them. And you can see it's not perfect, but you can still see both spikes and subthreshold voltage fluctuations. And this last fact that you can see the subthreshold fluctuations is quite important for our ID, as we'll soon see. So here we're looking at just one neuron in the microscope, but you can just zoom out and get a wider field of view. So this is the entire hole in the skull through which we imaged. The black lines are veins and arteries, and all the little white dots, each one is a neuron. So in movie form you get like a sea of shimmering lights. And note that you can actually see mostly only the cell bodies. So in the introduction slide you could see the wires, but that was because they inserted the protein into just one neuron. If you insert it in different neurons, then you cannot see the wires anymore. You can go and see the cell membranes. So you can't actually see the connections this way. You can only see the voltage of each neuron. So a step back. What does this all mean? We can now record the voltage of many individual neurons in living animals. And this is all quite recent, by the way. So this level of detail was not possible a couple of years ago. So that was voltage imaging. That's the context of our research, which is exciting stuff, but it's not what we do. We are computational neuroscientists. And we think that it's possible to use this new type of data for the problem sketched earlier. So we think that you could use voltage imaging data to infer connections between neurons, even though you can't see the connections. And because of the nature of this recording technique, you could follow the found connectivity over time without taking out the brain. So live connection detection. How would you go about that? Well, first, let's go back to the basics. Take two connected neurons. Measure the voltage in either. When the presynaptic neuron sends a spike, you'll see something like this. A tiny voltage bump in the other neuron, the postsynaptic potential. So there's this causal relationship between, on the one hand, two neurons being connected, and on the other hand, the spikes of one being followed by a little voltage bump in the other. And our idea is simply to reverse this reasoning. So if we see a spike followed by a bump, then maybe these neurons are connected, especially if this is a frequent occurrence. So the postsynaptic potentials. How are we going to test this idea? First of all, we don't actually need real neurons for our initial experiments. We can use simulated ones, so that we only need a laptop to work out this math for us. More concretely, we're going to model a neuron as a single point, which we give a voltage called V, and the time course of this voltage can be predicted using these differential equations that you saw earlier. We don't work them out by hand, we program them into the computer to let the computer solve it numerically. Next, we need to provide some input to the neuron, otherwise the voltage just stays flat. Inputs are modeled as spike times, or spike trains. We generate some random timestamps, which we say are the input spikes. If you run the code in the top right, it generates a graph like so. And note that every input spike corresponds to a little bump, so our postsynaptic potentials. If you get enough of these bumps, you get a spike. As nice as this is not the kind of signal that you would expect from real data. Because remember the voltage imaging setup, at each step of this path, various distortions are introduced into the signal. And to model this reduction in quality, we're going to keep it very simple at start. We just generate some Gaussian noise and add it to our signal. And if you then, you can tune the quality of your data by just increasing or decreasing the strength of your noise. So this is then our quote-unquote voltage imaging signal. So the stimulation from start to end looks like this. Your input spikes and your final output. Let's add some more inputs to the neuron. So we simulate a bunch of spike trains, some of them connected, some of them not. Now, how would you find out by just looking at the different input spike trains and the generated voltage imaging signal, which spike trains are connected? The simplest answer is windowing. So for every spike of a spike train that you're interested in, you take a window from the green signal. So you do it like this. And you take all these windows together and you average them. And the hope is that by doing that, you would average out the noise, and what would be left is, if the neurons are connected, is a remnant of your postsynaptic potential. And if they're not connected, you will not expect to see anything. So a little recap. Our test is simply a bunch of spike trains, some of which synapse onto one simulated neuron. We then cut out windows out of this voltage signal, this generated signal, triggered by the input spike times, and average the windows together. So what do these spike triggered averages actually look like? So again, because it's an average window, the time is now relative to the presynaptic spike, or the supposedly presynaptic spike. And if the neurons are connected, we get signals that look like this, which is indeed reminiscent of a postsynaptic potential. Now, how can we quantify the probability that this spike train is connected based on just this signal? Well, first we measure the STA height, of the spike triggered average height, and we plot it on a number line. Then we calculate a new spike triggered average, but this time based on random spike times. We get these random spike times by shuffling the real spike train. And if you then plot the heights of these STAs based on random spikes, you get they fall somewhere on the number line here. And so that then you can use this to actually calculate the p-value. And the limit of this p-value is defined by how many shuffles you do. In this case, we did 100 shuffles. So this is the case if the spike train is actually connected. If your spike train is not connected, your STA looks something like this. And same tests. Well, we don't expect any bump, and in fact we don't get any either. So we were quite happy with these results, and we wanted to see how far we could push it. So here we added a ridiculous amount of noise to simulate a very low quality voltage imaging. And if you make a spike triggered average window from this, you can still distinguish the postsynaptic potential. But here in this extreme situation, it becomes useful to have a statistical test, to which you could add a significance level to determine whether a connection is real or not. So at a 0.05 significance level, for example, we would indeed classify this pair as connected. So we can now make a very bold claim. Our idea works and the method is robust. Of course, this comes with an enormous if and but, which we'll get to in a second. But first, a quick recap. So, we want to follow connectivity throughout an animal's life. There's this new powerful technique that gives us voltage traces of different neurons in live animals. Our idea for connection detection is based on the causal relationship between the connectivity on the one hand and activity on the other. So when neuron A and B are connected, then when A sends a spike, it will generate a bump. So if we see a spike and a bump, then we can infer that A and B may be connected. We test our idea in silico by using a simple network like so. A bunch of spike trains, some of which synapse onto our simulated neuron. Now, given only the green quote-unquote voltage imaging signal and the different input spike trains, can we know which are connected? Well, we use the simplest possible test by taking these spike-triggered windows, averaging them together, and the results speak for themselves. But now, the big but. This simple setup is exactly that. It's simple. So we have shown that it can work here, but imagine that some of the input neurons are connected to each other, that they're actual simulated neurons, that there are recurrent connections, that there are also inhibitory postsynaptic potentials, that all the neurons have different properties, different types of neurons. All of these things make it incredibly more difficult. So that is exactly our next step. We are going to make the experiment more difficult and hence more realistic. So I would like to wrap up with a take-home question. If you could know the wiring before and after, which experiments would you do? Right, and with that I would like to thank you for your attention. You can find me online over here and check out our lab website. We are based in a big office in the basement, shared office, all the computational people, so come and say hi when this is over. And finally, thanks to the conference organizer, the tech team, and our chair Nick Myers. Back to you.